Yeah, but I'll look into it. Very curious. Yeah, okay. So now let's do genetics. Should have my PowerPoint up. Genetics. This is Gregor Mendel, Austrian monk and scientist. So Mendel lived long ago. He's called the father of modern genetics. And his studying of these garden peas, he studied heredity, was so foundational in our understanding of how genetic information is passed on from parent to offspring. And, you know, he didn't have Netflix. He didn't have basically anything to do. So he stared at garden peas all day and noticed their traits and how they differed over time. And depending on parent trait versus the offspring of the parents being these pea plants. And we're going to take a look at some of his profoundly important foundational theory of genetics. It's true. They became scientists because they were really bored and it was the only fun thing to figure out. It's very true. Okay. So get my notes up here. Before Gregor Mendel, this picture is actually from a very ancient history textbook. And this is actually what they thought of how humans reproduce or how genetic information is passed on. They thought the sperm cell had a mini human in it that would then later grow inside of the female. This is wrong, of course, but it's kind of interesting. So this was actually 1850s. So right before Mendel's time, excuse me, and his work sort of disproved this idea. What he really discovered is that both of the gametes, sperm and egg, contain genetic information that when combined are the toolkit or are the blueprint, really, the instructions to create that organism. Both sperm and egg have these genetic instructions. And of course, that's in the form of DNA, the chromosomes. He didn't know that. He didn't know that. It was only when technology became better and the structure of DNA was elucidated, that was found out later. This kind of unit with genetics, we also call it patterns of inheritance, because that is really what Mendel's famous for. These patterns of how certain traits are seen in generations or not seen following, okay? But let me show you what he was doing. So what he was doing here. So he would take to crossbreed these pea plants, right? He would go to the flowering part of the plant. So interestingly enough, plants have sexual organs and they produce sperm and egg, actually. I think most plants, remember, I'm not a botanist, but many plants are, what's the word? Hermaphroditic, I think. Not ambidextrous, that's left and right handed. But they have both male and female parts. So for instance, with flowers, the pollen that's produced in this part of the plant actually contains sperm cells. It's the male part of the plant. And so he would put a paintbrush against the pollen and get it covered in pollen. And then he would, to breed the pea plants, he would go to a different plant and attach the pollen with sperm in it to the female part of the plant. And actually, it's actually called an ovary here, the base of this part. So this pollen travels down, and inside the ovary of plants are egg cells. And so he's fertilizing these egg cells with sperm cells. I'm actually going to draw something that I think is really important. It's a very simple picture, but I really like to emphasize this, that in sexual reproduction and creating a new being, whether it's a plant or animal, anything that reproduces sexually, the sperm cell with 23 chromosomes combines with this much larger egg cell that also has 23 chromosomes. Now, and we just learned how that's made, right? Whether you're male or female, what you're making through meiosis. They combine to form the very first cell, which has 46 chromosomes of this new organism. So in Mendel's case, he was producing seeds to plant by crossbreeding these pea plants, and he would plant these seeds and note their characteristics. Yeah. This is actually why our somatic cells have 46 chromosomes. Combine 23 and 23 and you get the 46. Okay. So Mendel's producing pea plant babies by combining sperm and egg. Pollen contains sperm. Ovary of a different plant has the egg cells in it. Okay, so now we're going to do a bit of drawing. Sort of his historical landmark finding / experiment. This was in the 1860s, and we're still teaching it. That's how important it was. Okay, so let's do this. So we're going to start with the P, the parental generation. And I'm going to use my black here and my purple to represent purple flower and a white flower. So let's pretend we're Mendel. And we call this the parental generation, right? See what time it is. Oh, we're doing great, so I can slow down a little bit. Oh, they got pizza. My neighbors got pizza. Dude, they got 2 large pizzas. They can't eat that by themselves. I should knock. Sounds so good. Even though I had pizza last night. I feel like a burrito tonight. Anyway, back to Mendel. Or maybe I'll go to Walmart and get a bunch of Pop-Tarts. Oh, man. All right, so parental generation. He started with a flower pea plant that had purple flowers, okay? Bred that, crossed it with a white flower with white or lack of pigment petals. So here, let's do the purple. Yes, that's how I draw my flower. And he's crossing it with this white flower, purple and white. This is what he saw. We call the first generation in one of these experiments F1. F1 generation. This is the first generation. Mendel saw 100% purple flowers. No matter how many plants he would make, or no matter how many times he would cross a purple flower with a white flower, all their offspring, all the flowers, all the seeds that were produced gave rise to purple flowers, no matter what. And he planted a lot of flowers, like 600. He tried it with a ton of times he would try this and he would always get purple. So let's write 100% purple offspring. Of course, this was perplexing. This was very odd to him. Why? Because there's a white flower parent. He said, where the heck did that trait go? Okay. 100% purple flowers. F1 is first generation. So I'll write that in. First generation. These are the children flowers. Okay. F1 is 2 for this example, yeah. But I'm just saying, in general, when he would cross a purple flower and a white flower, he'd always get these purple flowers whenever he did this. But we're gonna, for the next part of this setup, we're gonna take 2 of them and actually cross those. So basically what's going on here is Mendel was crossing these, he was getting purple flowers. I'm gonna take 2 of those purple flowers and cross them. But again, I just want to emphasize in this first generation, he wasn't seeing flowers with the white pigment. And he was like, well, what's going on? Where'd it go? So he crossed 2 of these purple flowers. And in our F2 generation, which is our second generation, on average, Mendel saw, okay, Mom, don't text me right now, I'm teaching. On average, Mendel saw 75% purple offspring. So 1, 2, and 25% white flowers, white flower offspring. So I'm going to write 75% purple and 25% white. Heck, I might even just color these in just to emphasize here. Okay, so he thought this was so bizarre. I would too. Because he's crossing purple flowers and yet he gets some white flowers. But when he crossed the purple and a white up here, no white flowers, right? So something weird was going on. And so what's happening is we see the disappearance of this trait in the F1 generation. And it reappears in the F2 generation. So the F2, white, let's write white trait reappears in our F2 generation, right? So white trait reappears. Okay. I really wish I could leave this up because we're going to come back to it, but I only have one whiteboard. I can just quickly redraw it later. Let me set it aside for now. Okay. Oh, I'm sorry, I missed all the questions. F1 is 2 purple flowers. Yes, true. Although he would always get purple. I just took 2 of them and crossed them. Are the white and purple flowers at the top parental or F1? Parental. Exactly. They're the parental generation at the top. And then F1 are the 2 purple that came from the parental. And then the F2 are the 4 down here. What does it say above 100% purple flower? The white trait reappears. And I drew an arrow from the parental generation, the white flower up here, to the F2 generation where the we saw the white flower trait reappear. Because it disappeared in the F1, right? We only saw purple. Okay. Okay. So this was Mendel's theory. Ooh. Mendel's theory, or Mendelian genetics, as we call it. Each of our physical traits, or any organism's physical trait, is encoded for by a gene. A gene is a segment or a sequence, usually could be anywhere from 1,000 to 10,000 base pairs, nucleotides long in the DNA. So these genes in our DNA encode for these physical traits. The next, after genetics, we're actually going to learn how DNA codes for traits. Like why, how, and why, both. But noticing that these genes were heritable, they were passed on, right? So these traits were passed on to the offspring through DNA. He didn't know that. Again, he didn't know it was the DNA that doing this. I think he called them heritable units or something. He had no idea what the genetic material was of DNA. Now, the second important thing here, and this actually really connects to our discussion on meiosis a bit, but each gene is inherited as 2 versions. Okay, but I'll keep reading before I talk. So these versions, we call them alleles. And you get one version of the gene or allele from mom, and you get a different or the same, could be the same version of that gene from dad. So you're going to get 2 alleles for every gene. Okay? 2 alleles for every gene. Let's do an example. Well, we kind of just did, but we're going to kind of go a little deeper with what alleles are. Okay, this actually relates. This is why I like to introduce. So this isn't entirely like, whoa, what the heck are you talking about? When I was talking about ketchup, Heinz and Hunt's, this is it. This is what I was referring to with homologous chromosomes. And hair color. These are genes. And there were 2 versions I gave you. You could have the brown hair gene or the blonde hair gene, right? Next time we'll talk about how that can get a little more complex. But this is traditional Mendelian genetics, where every gene has 2 versions. And we just did an example of one. So the example was the flower color gene. The flower color gene had 2 alleles. at a purple allele and a white allele. Two versions of flower color, right? So I'm going to write purple, the purple allele, and I'm going to actually denote it with a capital P. I give it a tail because a lowercase p that I'm going to use for the white allele looks the same if you don't put the tail on it. But this will make a lot more sense when we do our next slide. But our purple allele, I'm going to just say it's a capital P. That's the allele for purple. The lowercase p for white allele. Many of you who have taken biology before, I'm sure dominant and recessive, these are familiar terms. But of course, this is introductory and I kind of, I teach everything. I don't assume that you know this. Okay, so we're going to talk about that. Also, very important. If you write allele in cursive, it's really fun. Watch. Anyway, how I figured that out, I think I was just really bored one day and just writing allele in cursive, and I was like, whoa. Okay. All right, so I've told you before, biology, new language, a lot of new terms, a lot of new words. We're going to learn some new words. Okay, the first is the genotype. So the genotype is the combination of two alleles for a trait. Okay. So if we have our two alleles in our example here, big P, little p, right? A genotype would be the combination of two alleles. How many combinations can I make here? And remember, I can have two of the same allele. Careful. So 4, technically 4. So we can show you why it's not 4, but it's very, you're on the right track for sure. So you can have big P, that's 1. You can have big P, little p, so I'll separate them with a semicolon. Can have big P, big P, little p, little p, and then little p, big P. But big P, little p, and little p, big P are the exact same. They're the same genotype. So we never do that. We never put a lowercase one first and then an uppercase. So I'm just going to get rid of it. So these are 3 unique genotypes now. So we have 3 possibilities. Now, we're going to talk about what this means, but if you have what it means by dominant. If you have an uppercase capital letter as an allele, it means it's a dominant allele. So this is dominant. If you have a lowercase letter, we call that recessive allele. So this is recessive. 10 minutes. Do we have time? I think we can finish or get close to finishing. That would be epic. Why is that determined with whether or not it's upper? You'll see once you learn what a trait that's dominant actually means. I don't know, biologists, the nomenclature, the way we write it is uppercase letter. You'll see it's easier to see and do these, what we call Punnett squares. Maybe some of you have done that before in the past, Punnett squares. Okay. So we learned what genotype is. It's the combination of two alleles. We got 3 genotypes, right? Up here. There are 3 only options. And we're gonna look at that in a minute. Actually, should we do that now? Now let's do phenotype first. Okay, so there's this thing called phenotype that depends on the genotype in the sense of your genotype determines your phenotype. What's a phenotype? The physical appearance. It's your physical traits, the characteristic that we see. So my phenotype for hair color is brown. Maybe my genotype is like a B, big B, little b, or whatever, right? Those are genotypes. The genotype will determine what your phenotype is. Okay, and we're going to look at what that means. But first, there's 2 rules. 2 rules to that. I just added this to the slide, so if you guys want to write it down, I'll just kind of chill while I read about it. But you can have a dominant phenotype, meaning you're going to see the dominant phenotype if you have 1 dominant allele in your genotype. You only need 1. And we're going to look at genotypes again in a moment. But if you have 1 dominant allele, 1 capital letter allele, you'll see that dominant phenotype. And this is in your genotype, okay, for that gene or trait. On the other hand, the way that you're going to see a recessive phenotype is if you have 2 copies of the recessive allele. If you only have 1 copy of that recessive allele in your genotype, you will not see that trait observed, that phenotype, whatever the recessive trait or allele codes for. You need 2, no dominant trait alleles. So we're going to do an example, or 3 examples really, of this genotype-phenotype relationship. So I think I'll erase this. Red hair is dying out. You guys are asking great questions tonight. I've never thought about this. It might be. I think red hair is rare for sure. It's more rare. And that means the combination of genes, it can get a little more complicated than Mendelian genetics, like 2 alleles. We're actually going to address it a little bit next time, but it's probably getting, I get what you're asking. I believe that most of the genes involved in the human race are dominant pigment genes that will overshadow red hair genes. Yes, I think it relates to this. And that's why if someone has red hair and another person has red hair, that offspring will probably have red hair because there are no dominant genes present or alleles. So I think it's a great question. You're definitely thinking about it in the right way. Oh, okay, sweet. Yeah, definitely thinking about this in the right way. Okay, so let's learn some more fun words. Heterozygous, homozygous recessive, and homozygous dominant. Yes, they sound all fancy and weird and whatever, but we actually just learned them. The 3 possible genotypes. And we're going to do it for this example of flower color. So let's make a little table. So heterozygous, actually, let me see here. Yeah, for the sake of time, you got just, it's on the slide, but the first one was heterozygous, right? Heterozygous was our big P, little p genotype. So a heterozygous genotype is big P, little p. Remember, I put the tail on it because they kind of look the same. And so this is one copy of the dominant allele in this genotype and one copy of the recessive allele, right? Homozygous recessive. That's 2 copies of the recessive allele, little p, little p. So in fact, actually, I'm going to write genotype up here. So these are genotypes, right? And then we have one more option, not 2. Homozygous dominant, big P, big P. So these are possible genotypes for flower color. 2 dominant alleles, 2 recessive here, 1 recessive, 1 dominant. And here's the interesting thing that occurs when we look at phenotype. Our heterozygous genotype here will give us a purple flower. We see the dominant phenotype. The dominant allele overshadows the recessive allele. So we see the dominant trait in a heterozygous genotype. The only way you see that recessive trait is if you have homozygous recessive genotype. So this would be white, phenotype. Physical characteristic, right? Phenotype. And then homozygous dominant genotype, of course, you're just going to see the dominant trait, which is purple. Okay. So really the last thing I want to do today, because we're not going to finish, but that's okay. There's only really a fun little practice, 2 fun practice things, problems or whatever that we can look at next time, which is totally chilling. We made a lot of progress today. The last thing I want to do is look at our original that you have in your notes. I'm going to redraw it. And we're going to assign genotypes to each flower in Mendel's traditional experiment, in his classic experiment. We're going to assign genotypes, and we're going to see why the pattern of inheritance works like how we saw it, where this trait vanished in the F1 generation and then came about again. The genotype thing explains why. So I'm going to redraw it, have our parental generation, our F1, and our F2. It looked like this. Okay. So with our final minute here, I'm going to go ahead and tell you the genotypes of each of these flowers. Oh, I forgot one. First of all, these are crossed, and then we're crossed another white up here. The genotype of this purple flower in the parental generation was big P, big P. Homozygous dominant. The white flower, I'll just draw it to this, and I'll draw it inside, why not? Little p, little p. The F1 generation, they were all big P, little p. So you only saw the dominant trait, which we said was purple. You never saw the genotype for homozygous recessive. Okay, and then the 75% that were purple here were either big P, or big P, little p. And then this one was little p, little p. Now, this explains why you saw the disappearance of that trait in the top generation, in the first generation. Wait, the disappearance of this trait in this generation. I know I'm over time, so I'll just say last. The offspring genotypes can be predicted by using a Punnett square technique, which we're going to practice next time. So I'll just write in predict offspring genotype using Punnett square. Which would tell you that if you cross these two flowers, you're going to get a 75% chance. 75% chance purple, 25% chance white. What square? Punnett. I'll type it into the chat, actually. Punnett square. And we're going to practice doing a Punnett square next time. Actually, a few practice problems. And I think the lab as well, you're going to. So it might be easier to start the lab after next lecture, after we practice that on Wednesday. But you can certainly do the meiosis portion of the lab for sure this week. That's it. I know I went a little over, but we made a lot of progress, so I'm pretty stoked. Okay. Sana, so first let me answer any lecture related questions and then we'll do other questions. Any questions about today's lecture, genetics or